Welcome to the first episode of Applied Biotechnology Advancement on the topic of COVID-19 and medical biotechnology. To start off with the table of contents, first with an introduction giving some statistics on the COVID-19 compared with other pandemics at the 1957 pandemic of the H2N2, moving on to the structure of the SARS-CoV-2 in order to understand its pathogenesis and its mechanism of action, as well as the modes of transmission, moving on to the medical biotechnology preventive means, focusing on how to develop new drugs to target the COVID-19 and add on what aspects should we consider, finally ending with the references. To start off, you see the COVID-19 is considered to have the highest mortality rate, having ranging from 0.15 till 0.28, which is the highest mortality rate since the 1957 H2N2 flu. The highest one was the flu of the 1918 of the H1N1. Also, you see the global deaths considered to have the highest also of 12 million, range between 12 million and 22 million, which is the highest also since 1957. Still not compared to the 75. Of course, all the numbers you see are estimated as adjusted to the 2020 population. The mean age of death in the COVID-19 ranges between 73 and 79. Which indicates that it's most lethal to elderly and immunocompromised individuals. So first, the structure of the COVID-19 is considered as an RNA virus, a single-stranded RNA virus having two main genes, the ORF1A and the ORF1B, both coding for non-structural genes. However, the structural genes are coding for the nucleoplasmic protein, the membrane protein, and the spike glycoprotein, or so-called the S glycoprotein, which is divided into two subunits, the S1 subunit and the S2 subunit. The mechanism of action starts by the S glycoprotein binding to the ACE2 receptor while the virus enters and the membrane fusion and RNA release occurs, followed by the translation using the ribosomes, producing polypeptide chains, which is then the RNA replication started via the viral polymerase. Of course, all these actions occurs inside the cytoplasm where the structural proteins are synthesized, and they are then transferred to the endoplasmic reticulum, a Golgi compartment, for the final encapsulation and the new viral release. The modes of transmission are simple. You see, the COVID-19 requires some kind of droplets of water in order to be transported. It could be transported in an airborne mechanism, inhalation, direct contact, or indirect contact via the mucosal or the respiratory systems. In medical biotechnology preventive means, we mean to focus on something from the essence of biotechnology. But first, as you see, the nefamostat antiviral, it works by binding to the S-glycoprotein, producing a competitive inhibition mechanism between the ACE2 receptor and the S-glycoprotein, which is a way that most of the drugs in the market at the moment are targeting. But we want to focus on some other aspects and some other options that could be considered in combating the COVID-19, as the sulfated polysaccharides, such as the carrageenan and the fucorin, which are extracted from algae. You see, it works by 4 different mode of actions. First, by blocking the viral attachment to the host cell, through the ionic interaction through the positively charged sulfated polysaccharide and negatively charged S-glycoprotein. The second mode of action by inhibiting virus penetration via disruption of the electrostatic interaction between the virus receptors and the host cells. The third mechanism of action is by inhibiting the virus internalization and encoding through binding to the allostatic site of the virus capsid inside the host cell, which in turn prevent its encoding. And lastly, through inhibiting the virus transcription and replication process through disruption of some replication enzymes such as the reverse transcriptase enzyme. Thus, the mRNA is not translated to the protein. Which brings us to the cherry of the pie of the SARS-CoV-2, which is the mRNA vaccines. The mRNA vaccine is defined as a vaccine containing the messenger RNA for the S glycoprotein. When it's injected, the mRNA antigen is uptaken by antigen-presenting cells, causing trafficking at the lymph nodes and priming of the T lymphocytes, which in turn activates the CD4 and the CD8, which in turn also start the germinal cell reaction and thus the immune response. These are some of the selected references. Of course, stay safe and thank you. If you have any questions, we'll be honored to receive your questions and comments. Please don't forget to leave your comments if you have any. Thank you.